The main focus of the research, though, is going to be with this microscope. And the plan there is to build up to detecting things directly inside cells at room temperature. This is the Institute's work. We now have a molecular biologist, and his primary task is going to be identifying which are the best targets, like basically which are the proteins that we expect to be involved in the magnetosensing. The optics guy, magnetics guy, and Clarice Yellow, the original founder, are going to be constructing the microscope. The microscope is going to fire a laser, bounce it off a bunch of mirrors, and then it's going to hit the target. The target then fluoresces, and we'll have nanosecond resolution in a single photon detector to get the fluorescence pattern. If all of this is right, the fluorescence pattern should contain a particular little oscillation called a Brabby oscillation, but basically it'll just look like a little squiggle. And then measuring the length of that is going to give us how long the superpositions are lasting in the relevant target. We're going to start with proteins outside cells at room temperature, and then we're going to look at proteins inside a protected environment inside cells at room temperature, and then just proteins inside cells at room temperature. As we do that, we're going to end up looking at sort of easier cases where we're going to have a better chance of seeing the mechanism.